Okay, well, you've just had John. It's me, Kathy, and welcome to Mindset. We're going to be working through life sciences now. Give us a call, 0861 058 262, or email helpdesk@learnextra.co.za. And then, of course, you can do Facebook as well, which is facebook.com. and then /LearnExtra. All right, I've been given a question here, which is on the board already, which says, study the diagram which represents the process of protein synthesis and answer the questions that follow. All right, everybody seems to battle with protein synthesis. Now, before we even go on to the question, I need to just show you a couple of things. And these are things that, if you know them, they're actually going to make your life a lot easier. When we look at protein synthesis, why and what is protein synthesis? All right, so synthesis means to make. Okay, proteins, everything, every cell, because remember cell membranes are made up of phospholipids. Okay, you've got your protein channels in the actual membrane. So every cell, every organelle in a cell, every antibody, every enzyme, all of them are made of proteins. All your hormones are made of proteins, okay, except your sex hormones. Your sex hormones are made of, so estrogen, progesterone, and testosterone are made of fat only. But every other hormone in your body is made up of protein and fats. But if you don't have protein, you can't make fats. Think about baking a cake. I can't bake a cake if I don't have flour, for example, unless you're master chefs and you're going to use eggs. But I mean, let's think of a normal cake. So to make protein, all right, we need carbons, hydrogens, oxygens, and nitrogens. We need that combination. Proteins, remember, are made up of amino acids. And amino acids are joined with peptide bonds. So each amino acid is going to have a code, and that code is made up of a triplet nitrogenous base. In other words, if we look at a normal the normal processes, we have 4 nitrogenous bases. There are 2 groups. Group 1 is your purines. Group 2, pyrimidines. Okay? Your purines are your guanine and alanine. Okay? Pyrimidines are your cytosine and thymine. Now look here. This is for DNA. So in a DNA molecule, your nitrogenous bases, there are only 4. You get 2 groups, purines and pyrimidines. Your purines are made up of guanine and alanine. The pyrimidines are made up of cytosine and thymine. So pyrimidines has a Y, cytosine has a Y, and thymine has a Y. Now, instead of thymine, your RNA will have uracil. So uracil is only in RNA. So your messenger RNA, your transfer RNA, your ribosomal RNA will only have uracil and never thymine. If you see thymine, it means we're talking about a DNA strand. If you see uracil or a U, it means that you're looking at either messenger RNA or transfer RNA or ribosomal RNA. Okay, so looking at these 4 nitrogenous bases, and instead of uracil, thymine, you're going to have uracil. Now also, if you look at uracil, it's a U is actually a Y without the tail. So pyrimidines are cytosine and thymine, and then uracil, which is the Y without the tail. And if you remember it that way, you cannot go wrong. Now, very important is that guanine will always only bond with cytosine, and alanine will always only bond with thymine, okay, always, to make up DNA. So this is what you are going to write on your whatever sheet you've got, whatever exam sheet you've got, A to T and G to C, because that is your combination process. All right, now, last thing that I need to show you, because if you know what I'm explaining to you here, people, you can answer any question that they give to you with regards to DNA and protein synthesis and DNA replication. That entire section, what I'm doing here now, if you know this and you understand it, you can answer any question and get 100% for it. Now, if we look at our DNA strand, okay, and then we've got our messenger RNA. This is going to have the code on. And then we have our transfer RNA, which is going to be the anti-code on. Okay? This is the difference. With our DNA, I'm going to just take any triplet base. So let's say cytosine, adenine, and thymine. Okay, that is on my DNA strand. Remember, the DNA is going to have a complementary strand. Now, the opposite side of that DNA strand, the complementary base for cytosine will be guanine. For adenine will be thymine. For thymine will be adenine. Remember, A to T and G to C, always. All right, so that's what my DNA strand complementary base will be, but we're working with what I've written in white here. So the messenger RNA is going to now code on to our DNA strand. This is the DNA strand we are working with. It codes onto that DNA strand as G, but remember, there's no thymine in RNA, so it will be a U, and adenine, the complementary base to thymine, is going to be adenine. So that is my code on. The code on is always opposite. This is always opposite to the DNA strand. In other words, it is the DNA's complementary base. All right, now we look at our transfer RNA, and the transfer RNA is always going to be the opposite it's always the opposite to what the codon is. So there's your DNA strand. Your messenger RNA codes on, so it is the complementary base, or same as the complementary base of the DNA, but remember, it's RNA, so there will always be uracil and no thymine. Then we go to the anticodon. Now it's anti, which means it is opposite. So what is opposite of G? It's going to be C. What is opposite of U is going to be A. And what is opposite of A? It's going to be not the T, because remember it's RNA, it will be the U. So your messenger RNA and your transfer RNA, you're going to have uracil instead of thymine. Your messenger RNA is the codon to the strand, so it will be the complementary bases. And your transfer RNA is the anticodon. It is the opposite to your codon, which ends up being the same as your DNA, except the thymine is a uracil. And if you remember this, you can work out anything, any protein synthesis that they give you, you can work this out. Just remember, you have your DNA strand, your codon will be the opposite to the DNA strand, and it's RNA, so thymine will always be represented by uracil. And your anticodon, which is your transfer RNA, is always going to be the same as the original DNA strand, except that the uracil and the thymine will be replaced. Now, if I look at this here, this little anticodon, this triplet base here, will represent one amino acid. Okay, and then we have a whole chain of these, will give me a whole chain of amino acids. The amino acids will be bonded together by a peptide bond to form a protein. So this here is everything. If you understand what I've done here, you cannot go wrong. DNA strand, the codon, which is always this, you are transcribing. So let's find another color here. This is transcription. In other words, the messenger RNA codes for the DNA by giving a complementary base. So that's transcription. And then the transfer RNA, this interpretation of the amino acid by the ribosome, this is going to be translation. In other words, you have translated the combination of the nitrogenous bases, you've translated that into an amino acid. So that's translation. Alrighty, so let's go back to our question. And you'll see everything I've just gone through now will be here somewhere. First thing you do when you look at your question is you are going to look at what your labels are. So A will be the ribosome. Okay. B is going to be the messenger RNA because it's transcribing what is going on here. You've got transcript. No, what am I doing here? Sorry, this is the transfer RNA. Transfer RNA, and then you've got C. Oh no, I really am messing this up. C would be your what is being coded on, and D would be your amino acid. So this would be the process of translation. Okay, so let's look at our questions. It says identify organelle A, well it's the ribosome, and organelle and molecule C. Molecule C, it's going to be, it's translation of, and it'll be the protein. Okay, so this is going to then punt out the protein. And remember, between the amino acids, we are going to have a peptide bond. All right, and that's it. Or do I have another page here? Okay, I do. It says, if the sequence of bases on the section of DNA strand is, now look here, we've got GTA on the DNA strand, CGA, CAA, then write down the corresponding sequence on molecule B. So let's just see molecule B, all right. Now molecule B, remember, is your messenger RNA, so what will it be on molecule B? So now we're looking at the code on, and if we follow the rules that I had all the way down here, remember your code on is the complementary base that you would have of the DNA strand. So going through this here, the codon, if we have GTA, remember A to T and G to C, always. So the codon is going to be, for the G, it's going to be C. For the T, it's going to be A. For the A, it's going to be U. Why? Because you're looking at RNA. So here we're going to be working with what molecule B, and molecule B is my transfer RNA. So it's the codon. Then I've got complementary base for cytosine is guanine, for guanine is cytosine, and for adenine is thymine, but remember it's RNA, so it will be uracil. Then CAA, so cytosine complementary is guanine, adenine and adenine would be thymine, and thymine, there is no thymine because it's RNA, so it's UU. molecule C, and molecule C was our proteins or our amino acids. So this is the protein that we are churning out over here. It's going to be the same as it was here. So, but remember, you've got your uracil instead of thymine, so it's going to be the anticodon, and the anticodon is going to be GUA, GCU, and GUU. Oh no, what have I done? I've just done the opposite. Sorry. See, not focusing here. Let's just take that out. Let's make more space here. Let's go back to our pink. I need the sequence. It's going to be GUA, CGA, and CAA. All right, so all we've done here is substituted the thymine for the uracil because it is the anticodon every single time. Right, that's another mark there. So clearly they didn't think it deserved a lot of marks. Now, this one is adapted. These are all questions, by the way, people, that we're getting from on Facebook. So that's why they haven't given me the names here. But okay, we've got question 2.1. And this is adapted from September from the National Senior Certificate Paper 1, question 2. Something else I need to mention here is that you are going to find that the syllabus for your IEB learners and your National Senior Certificate learners, the syllabus is identical. You have to do the national curriculum statement. It is the national syllabus. It is the difference comes in with the way questions are asked, okay, and the way questions are dealt with. So that's the only difference. The content is exactly the same. So for life sciences anyway, so here we go. The table below shows some of the base sequences on molecule C that correspond with different amino acids. Okay, so molecule C, that was the codon, the anticodon. So we say, right, we have base sequences on molecule B and the amino acid. The base sequence at D changes from CUU to AUU. What is the name given for this type of mutation? Now, look here. We have the base sequence changes from CUU to AUU. So, CUU to AUU. So it goes from being leucine to isoleucine. The minute that happens, we've only changed one nitrogenous base, and if one nitrogenous base has been altered, it changes the amino acid. We call that a point mutation. Okay, it's a point mutation. Why? Because only one point has changed, and that one point has changed one amino acid. So it changes from leucine to isoleucine. Okay, how would this mutation affect the composition of the protein molecule being formed? Well, clearly, we are now going to have an altered amino acid in the sequence. Okay, and when we have that altered amino acid in the sequence, it will change the specific protein. Okay, now, if that change is detrimental, then the organism clearly is going to battle and it will stop making that specific protein. All right, or it's going to be making a protein that the body can't use. And then we'll have other problems that kick in there. If it is something good, the organism survives. If it is something that's bad, the organism will die. And if it's something that actually doesn't affect anything, we then call it a neutral mutation. But you must know the difference between a frame shift mutation and a point mutation. Point mutation is just what you see here. One single little nitrogenous base is changed. When that base is changed, it changes the entire triplet combination. And that combination affects the amino acid that is then produced. All right? And, okay, that was the end of that question. So I'll wait. They're going to be loading on new questions. So I think what we'll do is we'll go to the studio for an ad break, and I'll see you all in a minute.